Hello everyone, welcome to my presentation. My names are Anitha Philbert. I work at the University of Salaam, Tanzania. This is part of my postdoctoral work that is funded by CARTA, and the title of my work is Dengue Vector Distribution and their Infection Status in Selected Regions in Tanzania. So dengue is one of the vector-borne diseases that causes approximately 400 million cases each year. But the viruses are transmitted to humans by vector mosquitoes from the Aedes genera, and the two species, mainly Aedes aegypti and Aedes albopictus, are the main vector across the globe. But in Tanzania, we only have Aedes aegypti that breeds abundantly in many parts of the country. Aedes aegypti is also responsible for other arboviruses such as yellow fever, chikungunya, Rift Valley fever, Zika, etc., etc. Although in Tanzania we have had several dengue epidemics and other arboviruses such as Rift Valley fever, and we also have some few cases of chikungunya, but the abundance, distribution, and viral infection status of the vector is largely unknown. So this is the gap that we are set to address. We collected adult mosquitoes from regions which have had dengue and other arbovirus outbreaks in the recent past. So the collection were done in the following regions, which are marked in red in this map here. We collected the samples from Arusha, Tanga, Coastal Region, and Dar es Salaam. Dar es Salaam has always been the epicenter of the previous outbreaks. So the adult mosquitoes were collected using the magnet trap, carbon dioxide baited, and were identified morphologically, and only mosquitoes from Aedes genera were preserved for further virus detection. The morphological identification gave the following results. The collection was dominated by mosquitoes from Aedes genera, followed by Culex, Anopheles, and other species that couldn't be identified morphologically. So because we are interested with the dengue vector, we further characterized the mosquitoes from Aedes genera, and the morpho identification revealed that all the collection were actually Aedes aegypti, and the distribution of the vector across the sampling regions is as shown in this figure. High abundance was obtained from Dar es Salaam, followed by Tanga and the Coast region. The three regions, Tanga, Coast, and Dar es Salaam, are all located along the coast of Indian Ocean, and they have a similar climatic condition that favors breeding and proliferation of the vector. The screening of the virus gave an infection rate of 2%, and only from the samples that were collected from Dar es Salaam during the 2019 dengue epidemic. The correction that was done before, the mosquitoes turned negative for the virus. So what does that imply? This means that the reservoir hosts other than mosquitoes that maintain the viruses in circulation during when there is no epidemic, and that's where the mosquitoes pick the virus from and cause infection. So it would be interesting to find out what are those reservoir hosts for control purposes. And the high abundance was documented in Dar. Also, the viruses were collected for the samples collected from Dar es Salaam, and this corroborates with the previous studies and show that Dar es Salaam is a high-risk region for dengue and other outbreaks, other arboviral outbreaks. At the moment, there is no vaccine for dengue and the other arboviruses except for yellow fever. There is also no treatment for dengue and the other arboviruses. So the vector control remains the most promising approach. So we recommend the intensification of vector control strategies in the country for the control of dengue and the other vector-borne diseases. So this is the end of my presentation. I acknowledge CARTA for funding this study, and CARTA is funded by the following donors. So once again, I thank you so much for listening.